Hello everyone, welcome back to KaushalBioPrep. In today's video lecture, we are going to talk about another topic of animal biotechnology, that is the genetically modified organisms or GMOs. So, what are genetically modified organisms? So, these organisms can be plant, animal, or even microbes whose genetic material has been altered in a way that does not occur naturally by mating or by natural recombination. That means this is something that we do in laboratories. So we produce genetically modified organism by making some kind of alteration in their genetic material, which can be DNA or even RNA based on the organism. So there are genetically modified plants, animals, and even microbes. And why we do that? We do this alteration so that we can achieve the desired trait in the particular organism. So these alterations are done with the help of the genetic engineering or biotechnological techniques. Different techniques are there, and they will allow you to add or delete a specific gene in the genetic material, and so that you can modify the trait. You can even introduce some desirable trait or delete some detrimental traits if you want to. So they are basically enhanced organism. Now GMOs are also called transgenic organisms. Transgenic varieties you must have heard. And when genes from one species are inserted into another, then they are called transgenic, right? So you take gene from one species and you will insert it into another one. That is the transfer of the gene basically between two species. So that is why they are also called the transgenic organisms. Now the modification can be cross species bound. That means these modifications can cross species boundary. So cross species modification can be there. That means this is species 1, this is species 2. These are two different species, but you can take a gene from species 1 and you can insert it into species 2, right? And sometimes even the vice versa, from this species to this. So within species you can modify, and between species you can modify. For example, you can transfer a bacterial gene into a plant genome, right? So these two things are there. Now what are the basic characteristic of a genetically modified organism? So they contain a specifically altered genome. First of all, their genome has been altered, and this is done by combining genes that would not naturally occur. This is very important. You will not find these organisms naturally. So they have some alteration in their genome. And now second thing is they are regulated by the governmental bodies. For example, there is FDA, there is EPA, there is USDA in the US, and in India there is ICMR also, है ना, Indian Council of Medical Research. There is DBT. So there are different bodies which basically foresee what kind of genetically modified organisms can be produced, or what kind have been produced so far. So there are rule and regulation over these, because this is a kind of science which can be used in a negative way. So there is a whole government body which is regulating all these genetically modified organisms to ensure the safety to human beings, to animals, and also to the environment. Now there are certain controversies. Excuse me. Yeah. So there are some controversies which exist regarding the allergenicity, their environmental effect, the gene flow, the biodiversity concern. That means ऐसा नहीं है that they are altogether only beneficial. Everything has a pro and con, right? So there are certain theories that these organisms may have some negative impact on the environment. They have negative impact on the biodiversity or other thing, but these adverse effect has been conclusively proven for approved GMOs. क्या मतलब हुआ कि अभी जितने भी ये concern है, they have not been seen in the approved GMOs. That means that is why there is a regulation over the production of the GMOs, so that if there is some kind of adversity, that can be excluded. Okay. Now how they are produced. So production of the cloned animals, this is something which you may have heard in very early in your studies, because we all hear about Dolly the sheep. Now I will be including this example in coming slides. Now what is this? So the production of the cloned and transgenic animals employed advanced biotechnological techniques to generate animals with desired genetic trait. for research, agriculture and medicine. Now what does that mean? What is cloning? Cloning means that you are producing an exact copy of something. So cloned animals has been produced and the transgenic animals have been produced. Both things work simultaneously. How? Because these techniques that you use for production of these, they are the same advanced biotechnological techniques. How they generate animals with desired trait. Why we are considering cloned animals? Because in cloned copies you will get the exact gene, right? And you have your desired, this gene must be related with the desired trait and you will have the generation for it, right? Next generation whatsoever you produce. Now the very commonly used technique for this is the somatic cell nuclear transfer. This was one of the first technique. Now what was the process and what is the process? Because this is still in practice. So a somatic cell nucleus, there are two types of cell in the body. One is the germ cell and another one is the somatic cell. So germ cells are those cells which basically make your gamete, right? So they are the ones which will go into the next generation. Somatic cell will die off with the one generation. That means in your body the somatic cell will not be passed on to your offsprings, right? So a somatic cell will be taken and the somatic cell nucleus is taken from the animal to be cloned. For example, if you, this is animal A, you want to clone this animal, so you will take the somatic cell from this animal and from that somatic cell you will take the nucleus, right? So a somatic cell nucleus is taken and it is transferred into an enucleated egg cell. Now what is this? Egg is a germ cell. So in one organism you are taking the somatic cell, in another organism you are taking the germ cell. This germ cell has no nucleus because you have denucleated it with help of certain technique. So this oocyte doesn't have the nucleus. From here you will take the nucleus and enter into it. And now you know the nucleus is the one which contains the genome. So now this germ cell will have the genome of the somatic cell, right? So the reconstructed egg is activated to start dividing and developing into an embryo. Now the embryo is then implanted into a surrogate mother where it is developed into a clone of the original donor animal. Why donor? Because you have taken the nucleus of that donor. So jiska nucleus hoga, that characters will be present in the clone. But now because the nucleus was altogether taken from the somatic cell of one organism, from that organism all the characteristic will be present in the cloned one. So that is why this is called the clone of the donor. So this is the technique. Basically Dolly the sheep has been had been produced by this method only. So this is a somatic cell donor. So this is the donor. From donor you will take the somatic cell. Now you can see the diploid chromosome in the somatic cell. Secondly, this is the egg cell donor. So this is sheep 1, this is sheep 2. Another sheep is you are taking egg. Now egg cell has the nucleus. This is haploid chromosome, but you will enucleate the egg cells with certain technique. You will denucleate it. Basically you will remove the nucleus. Now the somatic nucleus is transferred into this egg. Now your egg cell is having the transplanted somatic nucleus. Again this is in the dish. You will stimulate this cell to multiply. Now the 8 days old embryo is taken and it is implanted into the another sheep, that is the surrogate mother. Now the surrogate mother will give birth to the lamb, and this lamb will has the exact genetic combination of the donor. So that's why this lamb is a clone of the donor. That is how the somatic cell nucleus transfer is conducted. Now this is the cloning. Now what is the journal workflow if you want to create a transgenic animal? So there are certain steps. Number 1, you will design and prepare the gene construct carrying the desired gene. Now this is very important because you will need a vector. You want to transfer a single gene or maybe more than one gene into the recipient, right? या host आप कह लीजिए. So this gene will go in certain vector. So this gene construct you will design it, you will prepare the gene construct, and this gene construct which is your vector, it will be carrying your desired gene, right? Secondly, introduce the gene into the fertilized egg. Now you will take a fertilized egg and then introduce the gene into the fertilized egg. Or you can do this embryonic stem cell, in somatic cell, whatsoever you want, using one of the above technique. That means you can use any of the given. That means you can either take the fertilized egg, you can take the embryonic stem cell, you can take the somatic cell, and you can just introduce the gene into that. Then implant the manipulated embryo into a surrogate mother. Then you will screen the offspring for presence or absence of the gene expression, because this desired gene is known to you, so you know its expression, a certain kind of protein or something like that. If that protein is there, that means the gene is expressing. And it can happen that all the offspring do not have that, right? Only certain offsprings have the desired gene, so they will produce the expression of that gene. So you will screen that and then further breed positive transgenic animals to establish a stable line. So you will breed those who are positive for your particular gene, right? So what is the difference? You can see, then somatic cell nuclear transfer, you are taking the whole nucleus, so you are making an exact copy of this particular tree in this case, right? But in transgenic animal, you are not taking the whole genome, you are taking a particular gene. That is why I am telling you this is transgene, matlab a single or more than one gene you have transferred. So you will be having that gene, and there are chances that this particular gene will not be expressed if it is not accepted in the genome. It is not hybridized in the genome, right? So there are certain things to it. Now what is the application of it? So reproducing genetically superior animals, this is the major thing. Even with the cloning, even with the transgenic, mainly with the transgenic, cattle and sheep and pigs with high quality meat, high quality milk, even more quantity of milk. The cattle produces more quantity milk, because that particular gene which is involved with that, you have either enhanced the expression of that gene, or you have just added that gene into the genome. Then there is preserving endangered species, yet you can do that because you will produce clone of that exact organism. So you can preserve the endangered species जो naturally breed नहीं कर पा रहे, right? You will take the nucleus from the somatic cell and then you can produce it. Then there is a producing animals from biomedical research, that this has been a practice since a very long time. So you produce animals like genetically engineered mice are there, then there are guinea pigs, right? So this thing goes way back when we started the biomedical research, like drug testing or certain gene expression. So these animals are basically produced with the help of the transgenics. Now first example is the Dolly sheep. That was the first mammal clone using the somatic cell nuclear transfer, which this is what I have shown you regarding the Dolly sheep. And then the cloning after that has been a practice in cows, goat, and so many other animals. Now if we talk about the production of the transgenic animals, so there are certain methods for that. Number one is the DNA microinjection, right? Then there is embryonic stem cell method. You take the embryonic stem cell. There is somatic cell nuclear transfer with transfected cell. Now what is this? This is something which is added to this. Then there is viral mediated gene transfer. Then there is sperm mediated. Now let's talk about each. First is the embryonic stem cell method. In this, the transfection of the embryonic stem cell with foreign DNA. So what do? You take the embryonic stem cell, and in these cells you will introduce the foreign DNA, the foreign gene which you want to express. This genetically modified embryonic stem cells will now be injected into the blastocyst. I hope you are familiar with the blastocyst stage. In development biology, we read all about these stages, right? After the cleavage, certain stages are formed. Blastocyst is one of those. Now, genetically modified embryonic stem cells which have the foreign DNA, they are now injected into the blastocyst, which are implanted into the surrogate mother, right? Now the mother will produce a chimeric animal. Chimeric matlab combination hai isme. They have the foreign DNA also. So offsprings are tested for transgene integration. There are certain reporter gene which will let you know that your particular sequence of the DNA or the gene has been expressing or not, right? So this is simple. You are just taking the embryonic stem cell, you are introducing a foreign DNA, and then you are injecting into the this modified embryonic stem cell. They are injected back into the blastocyst, which is implanted into the surrogate mother. Second is the DNA microinjection. Now DNA microinjection में क्या होता है? The direct injection of the desired gene construct into the pronucleus of the fertilized egg. So there is no need to take stem cells. You are directly injecting the desired gene construct into the pronucleus of a fertilized egg. यहाँ पे याद रखिएगा, you need to take a fertilized egg. Now the embryo will develop with incorporated gene and it is implanted into the surrogate mother, right? And they are extensively doing this thing in mice, rabbit, pigs. What do you do? Like you see, this is the second method which I was talking, the DNA microinjection. So the desired gene with vector, you're using certain vector, plasmid or something, and this is the pronucleus. So you are just inserting it, you are injecting the desired gene with the vector into the pronucleus directly. Then this ये fertilized egg में हो रहा है. After that you will implant it into the uterus of the foster mother or your surrogate mother, right? So offspring will be formed and offspring will be tested for the transgene. Simple, right? इसके अलावा what you do, the first one which I told you, recombinant DNA and the embryonic stem cell. You take your desired foreign DNA which has been transfected into the embryonic stem cell. Now these stem cells they contain your desired gene. These are transfected into the your blastocyst stage. Basically ये आपका blastocyst बना है, transfected embryonic stem cell यहाँ पे डाल दिए. Now the blastocyst being implanted into the surrogate mother. So there are two methods. There are two methods you can do that. Then there is somatic cell nuclear transfer with transfected cell. We have already read about that somatic cell nuclear transfer, but with transfected cell, what is that? So combines the cloning with genetic engineering. How you do that? So somatic cells are genetically modified in vitro. So you first take out the somatic cell from the donor, right? And these somatic cells now are modified in vitro. Then the nucleus is transferred. So first you are doing in cloning, what you do? You just take the somatic cell nucleus and transfer it into the surrogate. But now you don't do that, right? Now what you do? You take the nucleus but modify it. That means you are transfecting the nucleus with some desired gene, and then you are going to transfer this nucleus into the enucleated egg, and then further the clone will be born. Now the clone will have extra gene which you have transferred. Now that because of that gene, this is a transgenic element. So this is also a clone and it's also a transgenic element. So it improves the efficiency over the previously discussed the pronuclear injection alone, right? Even the success rate of the somatic cell nuclear transfer is more. Then there is viral mediated gene transfer. In this case, you use certain retroviruses or the lentiviruses. These viruses are used to integrate the transgene into the early embryos or stem cell, right? So in you are using the retroviruses as kind of a vector so that you can integrate your certain desired transgenes into the embryo or stem cells, right? So this is a very good method for a stable integration and expression, but size limitation is there because retroviruses and lentiviruses cannot take up a foreign DNA of large base pairs. So the inserted gene size is their limitation of this method. You can see over there. So this is the viral DNA, this is the capsid. So this is basically your virus, right? So you are using the virus as a vector, but you can see that the DNA of the virus, it is modified. So it is modified DNA is injected into the virus because you see this is the red one, this is the virus own DNA, and you have inserted a new gene which is green one. So the vector, which is basically a retrovirus, has the gene which you want in your which you want to transfer into the host. So here vector is binding to the cell surface. This is host cell. Now vector will be packaging into the endosome when it is basically this is a vesicle formation. This endosome will now break. Acid dependent endosomal breakdown is there. So the capsid is released. Now this nuclear portion, the genomic material of the vector, this is going into the nucleus of the host, and there it will integrate itself into the cellular DNA. This integration is there, the enzymes are there in the vector's genome itself, and now the host will produce the certain kind of protein which you want because you have integrated those genes with the help of the vector's DNA, but the vector is your virus, right? And finally there is sperm-mediated gene transfer. In this you will do what? Incubation of the sperm with foreign DNA. So you will incubate the sperm with foreign DNA, which is then used for the insemination. That means directly sperm will be incubated with the foreign DNA. Now sperm has the foreign DNA, then sperm is used for fertilizing the egg. So DNA integration during fertilization is there. This is the donor sperm. ये artificial insemination करवाया. तो what they do? This is the collection of the sperm. Ejaculate में sperm आ गया. Removal of the semen fluid. Then sperm and DNA are interacted, because sperm is kept in the presence of the foreign DNA. Sperm will take up sub DNA, and then artificial insemination is there, and the particular foreign DNA will express, right? So these are certain methods for the transgenic production. I hope you understand these methods. If you have any kind of question, you can write down in the comment section. Now finally, the genetic modified animals, I am giving you certain examples, but I will encourage you to find more examples, especially for animals, because you are going to be competing in a zoology exam. So focus on the animals more. So there are certain very commonly known organisms. For example, there is aqua advantage salmon, which grows very fast than the normal salmon due to a growth hormone gene from the Chinook salmon. So this is a comparison, but you just memorize that a certain kind of salmon is produced, which is called the aqua advantage salmon, which grows faster than the normal one, just because it has a growth hormone gene inserted into it. So it is a transgenic animal. Then there is glowfish, which is fluorescent ornamental fish, which is engineered with the jellyfish or coral genes for glowing colors. Then there is enviropig, which is engineered to digest phosphorus more efficiently than the normal one. So it will reduce the environmental pollution. Very, very important. Then there is GM mosquitoes. Oxytextrine is there, which is modified to reduce the reproduction and control mosquito-borne diseases like dengue and malaria. This is still in developing zone. Basically, this is a recent you can say area of research, right? Now there is genetically modified microorganism also. So E. coli produce the insulin. How it produced? Because you have inserted the particular gene. So human insulin gene is inserted into the E. coli for large scale production of the insulin. This is very commonly done. Then there is yeast producing hepatitis B vaccine. So the yeast, which is Saccharomyces cerevisiae, it is engineered and it produces a viral protein which is used for the vaccine. Then there is the genetically modified bacteria for bioremediation, which are modified to degrade the oil spill or even take up the toxic waste, even for heavy metal uptake from the soil. So these are certain examples. I hope you understand something from the lecture. If you have any kind of question or query, feel free to comment it down. And if you learn something, please like the video, share it with your friends who can be benefited from the lecture. And if you are new to the channel, don't forget to subscribe. Stay connected for upcoming lectures. I will see you all in the next video. Thank you.